Notes on mitosis and meiosis. This will be b5A, and we're going to set these up a little bit differently. So we're going to make a chart, and we're going to go from there on the chart. So this is for us. This is Unit 9. It's M&M, mitosis and meiosis. 220. What today is? What is today? 2-2. And then this is period 7. Say hello to the people online. Period 7. That was very nice and vocal of you. Okay, so as I said, we're making a chart, so this is going to be slightly different from normal when we take notes. So just kind of bear with me on this. And there's a lot to write on here. I'm trying to limit, believe it or not, I'm trying to limit how much you have to write. So hopefully that'll help. This would be a really good time to use more than one color. I'm using 2 different colors here. Well, 3 different colors. I have the black to start off with, but I'm going to use 2 different colors to work on. It's nice to put the mitosis all in one color and the meiosis all in one color. You guys have heard of mitosis and meiosis before? You may have heard it pronounced meiosis instead of meiosis. You're welcome to do that if it helps you remember, but in reality it's meiosis here. So we have b5A, and it says describe the stages of the cell cycle. And this is what will take most of the time here. Describe the stages of the cell cycle. Is that big enough up there? Yes, no? Yes, okay. Okay, so this first piece right here is what we're going to spend the most time on. Now, I'm going to put some of these pieces in parentheses because we're really, the first one, it says including DNA replication. And we're not going to really cover that right now. We're going to talk about it when we talk about protein synthesis because it actually makes more sense to discuss that at that time to explain how it works. Okay? So when we talk about DNA replication, we really have to talk about the structure of DNA and how it's built and all sorts of really intricate stuff, and we're not going to hit that right now. We're going to wait until the next unit, I believe is when it is. So including DNA replication and mitosis. I don't know why they wrote and mitosis because that's a given. When you describe the stages of the cell cycle, that is mitosis, a part of that. Okay, the other thing that I'm going to just briefly touch on, so I'm going to just put kind of dots over here, and the importance of the cell cycle to the growth of organisms. And I'm going to put it in lowercase here because it doesn't take much time to do this part. And the importance of the cell cycle to the growth of organisms. Okay, so let's be honest. The cell cycle is like the life cycle of the cell. The whole reason you go through the life cycle of the cell is 1, so you can grow more cells, and 2, so you can replace damaged or dead ones. That's the importance. I mean, that's what one sentence does the importance of the cell cycle. So the importance of the cell cycle, 1, so you can grow more cells, 2, so you can replace dead or damaged cells. That's it. That's why the cell cycle is so important to the growth of organisms. So no deep, dark secret there in any of that. The other teak that I'm going to throw in here with this is, again, really, really simple, B6G. We already talked about it a little bit when we talked about the evolutionary theory, and remember how we talked about more diversity means it's a more healthy environment, it's a more healthy organism, things like that. So B6G says recognize the significance of meiosis to sexual reproduction. The whole reason for the process of meiosis for sexual reproduction is to increase diversity. That's the key. The more diversity you have, the better chance you have of the survival of the species. So, for example, and this is kind of pertinent right now, my aunt died from COVID. Okay, she developed COVID, she died from that. She was old, very old, but she died from COVID. I've been exposed to it multiple times with all you guys and your families, and you're bringing stuff in, and I've never gotten it. And if I do get it, if let's say that I got it and I didn't die from it, what that means is we have similar genetics. My aunt, we're going to have some similar genes in there, right? But my genes are enough different, there's enough diversity that I'm not going to die from that. The whole purpose of having sexual reproduction is to increase the diversity, so that if one person gets COVID and dies from it, it doesn't mean the whole species population of humans dies out. Every one of them are different. I had Lincoln came in, and his mom and dad had COVID. They're feeling better now, but they had COVID. He didn't get sick at all. He never caught it. So genetically, he's genetically different enough from them that even though they caught it and they were sick for a while, he might have caught it, but he never got sick. That's the whole thing about diversity. You have a greater chance of survival of the species. Okay? Now, that doesn't mean you're superior genetically to other people. It just means maybe according to that you are. Like, for example, I told you that my husband's family never gets fever blisters, but I do. My family and I do. That's just a genetic difference. So if you're talking about fever blisters, his genetics are better than mine to fight off that particular virus, right? Yeah, so it just depends on what you're talking about here. So that's the kicker. This is the whole thing for sexual reproduction, and meiosis is the formation of the sex cells. So meiosis is where we're talking about, hey, we increase diversity, and we'll talk about that, but really that's the key. The whole thing for the reason for sexual reproduction is for greater diversity. If they just clone each other, then if you can kill out one of them with the virus, you'll kill out all of them with the exact same virus, right? They're clones. They're exactly alike with each other. Okay, so I'm going to get two colors. Let's say brown, and how about blue? All right, so I'm going with mitosis here. So at the top of this one, I'm going to put mitosis. So everything on this side I'm going to put in blue, and it's going to be dealing with mitosis, and on the other half of the page will be meiosis. So everything on this side of the page will be brown and have to deal with meiosis. Okay, so when I look at mitosis, the first thing I want to write down is that this occurs in all your body cells. In meiosis, it occurs only in the making of sex cells. Okay, so earlier I said sex cells and people just started giggling. It's like, really? You are out of middle school. Stop it. So we have a scientific name for sex cells, and that's called gametes. Those are called gametes. So if we say generically sex cells, we can call them gametes instead. So you're going to see some vocabulary. So here's your first vocabulary word, gametes. That's the scientific name for sex cells. There are two sex cells for humans. What are they? Two different names. What are they called? Sperm and egg. Have you heard of those words? You can actually say the word sperm and not get in trouble for it, okay? So that's the scientific name, sperm and egg. Egg has more than one name for it. You can call it an ovum, singular, or ova, plural. So you might see it starting with an O, ova, ovum. When they talk about the creation, your body making sperm or making eggs, they'll talk about it as spermatogenesis. They're making sperm, or oogenesis. Two O's, oogenesis. So they have some different words, but egg is typically the standard word that you'll use for that. In mitosis is normal cell division. Where the chromosomes of the mother cell and you know what? I'm going to get tired of writing out the word chromosomes, so pretty much from here on out, I'm just going to call them chromes, okay? Chromosomes, okay? So the chromosomes of the mother cell are copied and divided equally between two daughter cells. So male or female, whether you're a male or a female, they call them mother cells and daughter cells. That's just in general. Here's the key. So you have all the chromosomes, you make a copy of them, you divide them equally into two daughter cells. So if you started out with 10 chromosomes, the two daughter cells end up with 10 chromosomes apiece. So for humans, we start out with 46 chromosomes, you make a copy, you get 92, and then you split them in between two cells, you get 46 chromosomes again. And it's not just any 46 chromosomes, right? You have 2 of the number 1 chromosomes, 2 of the number 2s, 2 of the number 3s, 4s, 5s, and so on. Okay? If you don't have those numbers, if you don't have 2 of the number 1s, 2 of the number, then there are going to be some major problems. In most cases, if the sperm and egg get together and the chromosome number is wrong, in the vast majority of cases, there's miscarriage that happens. The baby doesn't even grow at all. The cells don't even form and start growing. There are a handful of genetic diseases where it's a chromosome number issue, and in that case, things like Down syndrome, trisomy 21, where you have 3 of the number 21 chromosomes. Yes, you guys have all heard of that. There's also Down syndrome from, I think it's 3 of the number 14 chromosomes, but don't quote me on that. But so there are different diseases. There are a few diseases that the baby actually is born where it has the wrong number of chromosomes, but the vast majority of them, if you mess up on the number of chromosomes, that's really, really bad. Okay, so on the meiosis side, this is normal cell division. So both of them are normal. Normal cell division, where the chromosomes, there I'm going to abbreviate it, chromes, of the mother cell are copied. So far it's the exact same, yes? And divided. Look at this. Normal cell division where chromosomes of the mother cell are copied and divided equally between 4 daughter cells. There's your number difference. So in mitosis, you produce 2 cells. They have the same number of chromosomes as the mom. In meiosis, you produce 4 daughter cells, and they're going to have half the number. And that should make sense to you, because when you make sex cells, each sex cell has half the number of chromosomes, right? So that when you have the sperm meet up with the egg, they each give half of the chromosomes, so that you create a new organism there with the full complement of chromosomes. Okay, in a human, so let's talk specifically for humans. So, different organisms have different numbers of chromosomes. The chromosomes, you talked about the hedgehog had something like, was it 90-something chromosomes? I can't remember on that. I can't remember what the number was, but it was quite a few chromosomes more than us. That doesn't mean they were long or as big as our chromosomes or anything like that. It just means there were that many separate types of chromosomes that are on there. So humans have how many chromosomes? 46. Human begins with 46 chromosomes. And remember, 2 of each. So again, 2 of the number 1s, 2 of the number 19s, 2 of the number 21s. You have 22, they're called 22 pairs of autosomes, so that's 44 autosomes, and then the last 2 chromosomes are special because they determine whether you're male or female, so they call those the X and the Y chromosomes. Okay? So of your 46, 23 pairs of chromosomes, one of those pairs isn't numbered. They're called X and Y. Yeah? Okay. And they can tell, you have to get a lot of practice in that, but they can tell which ones are the number 1 chromosomes, which ones are the number 7 chromosomes. It takes a lot of work to do that, but they can figure out what's what here. So they start off with 46 chromosomes. They're copied. Everything makes a copy, so it bumps up to 92. And then they're divided into 2 cells, each with 46. Yeah? So you start out with 46, you end up with 46. For meiosis, we're still talking about humans, right? So we begin with 46 chromosomes. They're copied, so it bumps up to 92, right? And then it's divided into 4 cells. Each with how many? So what's 92 divided by 4? 23 chromosomes. So again, that number only applies to humans. Different organisms have different numbers of chromosomes. They don't look the same at all, so it's just a different process here. Corn may have 9 chromosomes. You know, it just depends on what you've got here. Okay, so now I'm going to draw a line through here. Just kind of separate these things out. And from here on out, we're going to do the cell cycle. And I'm going to put a circle around this one just so it's different from the others. So this is all the cell cycle. Okay? If you look up on the board over there, up on the TV, it says mitosis, and underneath it says IPMAT. Those are the stages: interphase, prophase, metaphase, anaphase, telophase. I can guarantee you on the free response part of your test, it's going to say list the stages or phases of mitosis in order: interphase, prophase, metaphase, anaphase, telophase. Learn that, memorize it, be happy with it. On meiosis, basically it goes through things twice. So you start off with interphase, prophase, metaphase, anaphase, telophase, and then it hits it again: prophase, metaphase, anaphase, telophase. So it's got to do 2 splits. It splits it the first time around, and you have 46 chromosomes in each cell. Then you have to split them again so that you have 4 cells with 23 chromosomes in each one. And we'll look at a picture of that in just a little bit. Okay, so the first phase is interphase. And I'm going to put a box around this one, so all of them with boxes around them will be the phases of mitosis. In interphase, that prefix inter means between. And so I'm telling you basically how they came up with the names for these things. So when we talk about between, a lot of times they talk about, okay, it's between cell divisions. Okay, so it's not reproducing immediately. This is the time that it spends most of its life in. So this is when you have a little bit of time for growth. So right after the cells divide, they're in interphase, and then they're going to grow a little bit. They don't spend a lot of time growing. Your individual cells don't grow a lot. Most of the time when you grow, it's more cells that you add for the most part. Not perfectly like that, but more cells generally for the most part, not just your cells get bigger and bigger. If that were the case, you would start off with little microscopic cells, and by the time you were 10, you'd have big Godzilla cells that you could see without a microscope, right? So that's not what we're talking about. Typically when you grow more cells. They don't just get bigger and bigger. So once the cell divides, it'll grow a little bit in size, but not very much at all. The really important thing that we're doing here is this is when protein synthesis occurs. Making proteins, protein synthesis. And different cells are doing different proteins and stuff. So basically this is when the cell's doing its job. So it's just in interphase, it's doing its cell job. If it's a bone cell, it's making calcium phosphate. If it's a muscle cell, it's making actin and myosin proteins. If it's a epithelial cell, it's filling up with melanin and keratin and things like that. If it's a pancreas cell, it's either making insulin or glucagon. If it's a pituitary cell, it's making a lot of stuff, but growth hormone might be one of those. So it's doing its job. That's what it's doing when it's in interphase. This is where the cell spends most of its lifetime. For you, it'd be like, okay, so you have some time as an infant, you have some time as a little kid, you have some time as a teenager. Most of your life, hopefully, is going to be lived as an adult, yes? That's the vast majority of the years that you have. In this case, the adulthood would be like the interphase. You're doing your job, you're doing what you need to do, taking care of business and everything there. Now, at this phase, DNA is in chromatin form. So the DNA is in chromatin form. And I'm going to underline this. Here's another vocabulary word. So you had gametes up here, right? Gametes. And you have chromatin. Chromatin is loose, unwound DNA. So loose, and I'm going to run right across my line here. Loose, unwound DNA. It's not visible in the microscope. We're going to look at some cells next week, and we're going to look for ones that we can actually see the chromosomes in. The vast majority of cells that you see, if you see the nucleus, you can't see chromosomes in there at all. So in this case, this is loose, unwound DNA. So again, think of that plate of spaghetti, cooked spaghetti noodles. That's what it's like. It's like all kind of messy and all tangled up in the side there, and that's how it is. And it's such thin strands that you can't see it. So the vast majority of time, it's in the chromatin form. So it's loose, unwound DNA. In order for you to make proteins, you have to have the DNA unwound so that the RNA that comes along can read it and write down what it says. So there are two parts to protein synthesis: transcription and translation. Transcription means, hey, write down the message that the DNA has. The DNA is kind of like in a secret code. You know those letters A's, G's, C's, and T's? That's a secret code. The order of the letters is a secret code. And the mRNA comes along and copies that down, just a section of it, and takes it outside to do what it says, to figure out what the code says. So it's got to be loose and unwound so you can make a copy of something, to figure out what is it telling you to do. Towards the end of interphase, the DNA is replicated. It's copied. You make an entire new copy of it. So you're about to divide into two new cells, and those new cells both have to have their own copy of the DNA, right? So at some point, while it's loose and unwound, you have to make an exact copy of the whole long strand of DNA. Have to make that exact copy of it. That happens towards the very end of interphase. And then also during this time, towards the end of interphase, the cell checks the DNA for damage. That's just crazy. It's crazy that your own cell can do this. And this is where a lot of times your cell picks up and says, hey, that's wrong, I've got to fix that. Now, if you were born with some genetic malfunction, something genetically wrong, a mutation already, and so all of your cells have that messed up DNA, it's not going to fix that. All it does is it looks to see, is this copy the same as the copy I just made? So it checks it for correctness. And most of the time, it catches most stuff, which is pretty amazing that it catches most stuff. Every once in a while, you get something that goes wrong, but the vast majority of the time, it's going to catch any mistakes that are in there. On the other side over here, let's just put same. So interphase is basically the same for both of them. Unless you want to write all that crap down again, I'll just put same. So during interphase, the cell's doing its little cell job, it's making proteins, the DNA is in this loose kind of spaghetti chromatin form, and it's making copies, it's making proteins, it's doing everything it needs to do. And then towards the very end of interphase, the DNA makes a copy of itself, the whole long strand, and it checks it for accuracy. So the cell's going to check all that stuff. Okay, so let's move on to the next one. Okay, so the next one we have starts with a P. It's prophase. Oh, sorry. Prophase. I'm going to put a box around it here. And then in meiosis, however, instead of prophase, it's called prophase 1. Because we're going to go through, if I can spell it, phase 1. So on the mitosis side, it's just prophase. On the meiosis side, it's prophase 1, because if there's a prophase 1, what else is there going to be? Prophase 2. Okay? So in prophase, the prefix, a lot of times we think pro and we think a professional, right? In reality, when we say someone's a pro, that's not using that as a prefix. It's just using it as a shortened version of professional. We just got lazy, kind of like I don't want to write out chromosomes, so I just call them crumbs. Write them down that way. You don't want to write out the word professional basketball player, so you say it's a pro basketball player. Yeah, same thing. In this case, however, we're going to look at the prefix pro, and the prefix pro is similar to pre. It means before. Okay? So in this case, it typically means the way they came up with the name is that this is before, these things happen before the DNA is ready. That's why they came up with this name, before the DNA is ready and in place and stuff. So that's where they're coming from with the name from that. Let me fix this so I can just a bit. Okay, so we have, let's see. Is that blurry or is that okay? Okay. All right, so prophase means before, it's before the DNA is ready. And this is when, during this time, the chromatin, remember chromatin is loose, unwound DNA. The chromatin condenses into chromatid form. Okay, really similar words. See that? The only difference is the ending on the letter. So here's another word that you need to know right here, chromatid form. All right, so we've done you a disservice in science because a lot of times we refer to chromosomes and we talk about in actually several different forms instead of being very specific. Typically, if you see them through the microscope, or if you see a karyotype where someone has them where they've taken a picture of the chromosomes and they put them in order, that's what we call chromosomes when you're just looking at it when people say, oh, those are chromosomes. When in reality, half, and you see them in the forms of X's, right? A lot of times they'll be two pieces together. Sometimes they'll be in X's hooked together. Sometimes they're smart and they pull them apart like this. Okay, so here's the deal. If it's in chromatid form, it means the original and its copy, and they're hooked together. They form that little X. Okay? One of those technically is a chromosome. The other one's a chromosome, but when you hook them together, you say this is a chromatid and this is a chromatid. We call them sister chromatids. They're kind of weird that way. So they're condensed into chromatid form, which means that, how do I say this? Let's say it means that you have the original chromosome and its copy. And they're hooked together. So we call each one of them a chromatid. But in order to be in this form, they're always going to be hooked together. They're always going to be in that hooked together form. If they're, do you guys know what a karyotype is? Yes, no? Okay, so when I want to see what all my chromosomes are, they find some cells in me that are going through mitosis. They take a picture of them, they cut out all the little pieces, and they line them up. So they'll have 2 chromosomes that are number 1s, 2 number 2s, number 3s, and so on. Yeah? Okay, so that's what they're talking about doing here. Now, when they take those pictures are actually the DNA that's in chromatid form. It's one chromosome with its copy hooked up to it. So when we say chromosomes, when you see the pictures, they're actually more than just a chromosome. It's the chromatid with its sister. Yeah? Okay, I know that's weird, but stay with me on that one, okay? So the chromatid is the original chromosome and its hooked together and its copy hooked together. So one's a chromatid, one's a sister chromatid. They're hooked together, yeah? All right, so the nucleus, during this time, the nucleus breaks apart. Because we have to get those chromosomes, that DNA, out of the nucleus. The spindle fibers form, which is weird. We'll talk about those a little bit later. Spindle fibers. Okay. Now, I'm going to go back and I'm going to put this other piece in red here. So when I talk about chromosomes, chromosomes are one complete strand of DNA. Can you see that? No, you can't. One complete strand of DNA, and it's condensed. It's wrapped up. When I say condensed, it's not loose and spread out, it's all wrapped up. So it's not the picture we see through the microscope, even though we slangily just call them chromosomes at that point. So there's a difference between what you're going to see in the microscope and what's actually a chromosome. I know it's kind of a stupid thing to say, but it's true that way. Okay? So one chromosome is one complete strand of DNA that's condensed down. So when I say you have 46 chromosomes, you have 2 of the number 1s, 2 of the number 2s. But when you see them in the microscope, you're seeing the original chromosome and its copy hooked together. Yes? You guys kind of following me on that? It's just a weird little thing. Okay, on the prophase 1 side, the things are going on here. So we have these things. The chromatin condenses. To form the chromatids. Okay, so you've got, when you say chromatids, you're talking about the original and its copy, right? The original and its copy, yes? One's the chromatid one and one's the sister chromatid. The original, they're hooked together, yes? But now, what we're not going to do, we're not going to leave them like that. It's going to be the chromatin condenses to form chromatids, so you've got 2 hooked together, so that's copy plus original. Yeah, sorry. Copy plus the original. And the homologous chromosomes. So it's hooked with its, and it pairs up homologous chromosome. Okay, so now you've got the original number 1 and its copy, and don't you have 2 of the number 1 chromosomes? So you've got the original number 1 and its copy, and then you have the other number 1 and its copy. So you've got 4 chromosomes hooked up together. Okay? So that means, when I say homologous chromosome, think about its mate. I think that's the best way. Its mate, its pair. If you think about a pair of socks, it's the other half of a pair. So you have 4 DNA strands hooked together. And we call that a tetrad. How many is tetra? Take a guess. 4. Yeah, tetrad's 4. The reason this is so important, you're thinking, holy crap, why do we have to know tetrads? I'm 4 of them, blah, 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 blah. The reason this piece is so important is because this is where crossing over occurs. And that's the official name for it, crossing over. Crossing over occurs. And that's when pieces of the chromosomes swap places. So it's swapping sections of DNA. That's just weird. But the reason it does this, and the reason this is so important, goes right back up to that b5G, 6G, where it says, why is meiosis so important to sexual reproduction? This increases diversity. This increases variations in an organism. So the parent is not going to be exact. There's no way that the parent can have a clone. It's not going to be exactly the same as the offspring at all. So it increases that diversity. How much time do I have left? 7 minutes? Okay, so here's what I want you to do. Once you've written that down, I want you to flip over to page 19 in your packet. And if you're online, I'll post this picture online so you can see this too. Page 19 in your packet. Okay, and I'm going to shrink this down some. Okay, I like this page. It's clean, it's simple, not a whole lot going on. This organism in this picture only has 4 chromosomes, right? 4 chromosomes, that's it. Do you see that there are 2 of the number 1s and 2 of the number 2s? 2 of the long ones, 2 of the short ones? So they're in pairs like that, right? So, and most organisms are going to be in pairs like that. Every once in a while, you get some that are odd numbers, but that's not the typical. So we have two that are alike and two that are alike. So when it goes through mitosis, here is, let's say here's the original on this side, and here's the copy that's hooked up to it. Yeah? So we have the original and the copy hooked up. So when this goes through mitosis, one's going to go to one side, one's going to go to the other. And so you have in each cell 1, 2, 3, 4, just like the original. They're clones of each other. Yes? Look at meiosis, though. Here we start off the same thing. But right here, when they're pairing up, notice you've got each set individually. And over here, you've got one, so you have the original and its copy. And then you have the other number one. Here's the other number one and its copy. I'm going to put polka dots on that. The other number one and its copy. And while they're situated like this, so these are all basically this, they have the same, they code for the same thing. So if the top of this one says, hey, you're going to have brown eyes, then the top of the next one's going to talk about your eye color, and the top of this one's going to talk about your eye color, and the top of that one's going to talk about your eye color. Okay? And what happens is when they're close together like that, they swap pieces. Like the tip of this one pops off and swaps places with the tip of the one next to it. So they rearrange genes on the chromosome, which is crazy. So you get this weird rearrangement of stuff here. That's what crossing over does. And it means that the original chromosomes won't be exactly the same as what you got down here in the babies. So look at this. In the first split, we separate these two to pieces. So look, now it looks kind of like this, right? A little similar to this. And then you separate them apart right down here, and so you're into 4 cells now. So we have mitosis, and we have meiosis going on in here. Yes? Okay. Let me stop capture here.